Dr. Genevieve Wojcik did her PhD at Johns Hopkins Bloomberg School of Public Health at Baltimore, and later she did a postdoc at Stanford University. She's currently an assistant professor of epidemiology at her alma mater at Baltimore, and she is interested in using genetics and epidemiology to understand the role of ancestry in genetic risk to elucidate the genetic susceptibility to disease and vaccine response. She's particularly interested in making an emphasis to develop solutions to address health inequities for diverse and mixed populations. So, Genevieve, thank you for being with us today, and we're really looking forward to your talk. Thank you.
Thank you for the great introduction. I'm sorry I couldn't be there with you today, and also that this whole thing will be in English, despite marrying into a Cuban family years and years ago. My Spanish is still abysmal. So I'll get started here. And so today, I think a lot of the things I'll be telling you might seem like common sense, I think, for people within the communities, but hopefully what I can show you is a framework to sort of work through how we conceptualize genetic risk when it comes to complex populations, taking into account genetics, environment, and genetic ancestry. Okay, so the bulk of my research is sort of emphasizing this main point that we have in the United States, and just sort of a disclaimer, this talk is going to be done from a very US-centric perspective. Some of the frameworks are applicable externally, but a lot of the sort of dynamics that I'm going to be presenting to you are very US specific. And this sort of premise is that, you know, the disproportionate burden of chronic disease in the United States is found in minoritized groups, yet our genetic studies are overwhelmingly done in those of European ancestries. And we can see this in the data. So what I'm showing you here is the GWAS catalog. GWAS are genome-wide association studies, so pretty much an agnostic look across the entire human genome to try to identify variants that are associated with various outcomes. And what I'm showing you here is the mean sample size for published GWAS, stratified by how the GWAS catalog categorizes these study participants. And what you can see is that in the last decade, so you can go further than this, but in the last decade, there's been this sort of really exponential increase, both in terms of the cumulative knowledge base, which you can see in the solid lines, as well as the annual mean sample size in the dashed lines. And you can see, particularly in the last 5 years or so, there's been a sort of exponential increase, primarily due to the UK biobanks, sort of large-scale biobanks, which have massive sample sizes. But if we sort of zoom in on the last 5 years for sort of everyone else, we're not doing so great. So here you're seeing pretty much the rest of the world. And what you can see here is that while there are some fluctuations by year, that what you actually see is a stagnation across the year. So we're not doing that much better. There's a lot more attention, I think, to diverse genetics, but we're really not getting the study sizes that we need for discovery, which is sort of the rate limiting step for genomic discovery. And so you can kind of think, all right, why is this sort of the way it is? And when we think about the sort of these end games, sort of the genomic health inequities that we see, and we go back step by step by step and try to find out where things went wrong, it sort of turtles all the way down. And by turtles, I sort of mean there's this Eurocentric bias that permeates every single step along the process. So if you start at the health inequities at the top there, and you go down just one step to translation, you can sort of see that translational work is often done in white populations, these white biobanks, where it's assumed to be the default. And then if you go back sort of one more step to the discovery stage of things, then what you can see here is that there are the out-of-the-box methods historically have been assuming a single population, a sort of single homogeneous population, which have been white and sort of European ancestries. And while this has changed somewhat in the last few years, it's not done as much to be the standard as what would be nice. And then if you go further back even one more step as to who is sampled, how their genetics is measured, you see more bias and sort of who is the default population. And sort of even in the technology that we use, there's been this bias that has been somewhat ameliorated over the years, but still present in terms of our reference genomes, et cetera. Okay, and so really what this all boils down to is that we have accepted as the default in these large-scale genetic studies a very particular population, which is mostly white, relatively healthy, well-to-do groups. And so this sort of has consequences across the entire pipeline. And so this sort of permeates into genetic risk studies. What I'm showing you here are polygenic scores. Polygenic scores are essentially one number that you give to a person that summarizes their genetic risk across the entire genome, whether that's hundreds or thousands or millions of sites. And they're typically built off of these genome-wide association studies. And so you can imagine that if you have a lot of bias in your association studies, then you also are going to have even more in your polygenic scores. And these are the scores that are being given back to patients, to customers, and these sort of direct consumer testing. And what you can see here is sort of by this past summer in the polygenic score catalog, which curates published scores. What I'm showing you here on the left-hand side is all of the scores that have either only European ancestry groups in the solid blue, or have mostly European and maybe some other groups added in. What I'm showing you in the middle is this is all the published scores, proportionally out of all the published scores, that include anybody who's been identified as African American, Afro-Caribbean, or Hispanic or Latino. And so what you can see is that it's a much, much, much smaller number of studies, and then even smaller number of studies that have only just like one or two of those, so more sort of cater to particular genetic backgrounds. And this is not representative of the global dynamics for sure, the global demographics. And what I'm showing on the right-hand side is it's definitely not even representative of the US demographics from the 2020 census. Okay, so why do we care about this lack of representation? One of the main reasons we care in terms of a methods development standpoint is that when you have a polygenic score, which is summarizing, again, the genetic risk across the entire genome, you're relying on a particular set of genomic backgrounds in terms of allele frequencies and linkage disequilibrium patterns. And so the further you get away in genetic distance from that training program, training set, the worse your risk scores do. And so what I'm showing you here is a paper that came out this past year from the Pisani group in UCLA, led by Yi Ding. And what you see here is each dot is a person, and you're seeing their individual level accuracy for a score as a function of the genetic distance. And they've separated people out into sort of European ancestry, Hispanic, Latino, South Asian Americans, East Asian Americans, and African Americans into these different colors. And what I think I really want you to get for this point, there's a lot to delve into, but the main point is that there is a sort of linear decrease in accuracy as a function of genetic distance. And this is seen both between the different groups, which is what we typically can think of, is that between groups we see differences. It's sort of a well-published phenomenon. But also within groups, you see a large amount of variation, where even if you are in a different group from the training set, if you're not that far genetically speaking from in terms of genetic distance, your accuracy is pretty good. But the further you are even within the group, the worse the accuracy is. So this is one of the big problems we have right now in the sort of delivery of polygenic scores to a patient population, let's say. And so it's well published, we know that PRS performs differently by racial ethnic groups. And my work focuses primarily on how this is complicated by recent admixture in individuals. And so can we see a difference within the self-identified group as a function of genetic ancestry? And then also, what happens when you compound that substructure within one group with environmental substructure as well? And so I think this has been much more well studied, more in ecological studies in terms of non-humans, but from the human sort of epidemiological standpoint, it's been a little bit understudied. And so what I'm going to present to you is sort of this epi way of thinking. So I come from an epidemiology background. It's what I teach now. So I think of everything in terms of these causal frameworks or potential causal frameworks. And one of the big concepts we have is that when you estimate risk for anything, genetic risk, whether it's risk for genetics or any other outcome, what you're doing is saying, compared to people who have every other factor the same, except this one thing I'm looking at, in this case, a polygenic score, this is the difference in risk, right? Which makes you have to have your comparison group be the same in every other way except for the one thing, which is the polygenic score here, and that's your counterfactual. And so typically when we think about polygenic scores and the relationship to outcome, a lot of methods have focused on really trying to cut off this one relationship between more the population genetic side of things, in terms of allele frequency differences, LD patterns, and then the polygenic score to make sure that we don't have any confounding or bias from these other aspects here. But the problem is that you have a lot of other things that are affecting both outcome and the score, because scores, again, are capturing correlation structures. And so if you fail to capture all of the heterogeneity in the entire system, weird things can happen and you're not really giving people correct estimates. And this is especially problematic when you're trying to translate things from a population level, which is most of these studies, to an individual level, which is a patient coming into a clinic. Okay. And so what I'm going to do here with you in the time that I have left is really build this picture, build out this causal framework with you using work that we've done both within my group as well as collaborators and then sort of the field at large. And so typically when we think of genetic studies, we really focus on the relationship between genetics and the outcome. So for this example that I'm going to walk you through, we're going to use body mass index, which is BMI, not because it's medically relevant, but because it's really an interesting example of a trait that is both genetically and environmentally influenced. And so if we think about this study between SNP and BMI, first we have to figure out, how does ancestry fit into this? We know that ancestry can influence SNPs due to allele frequencies, LD, everything, but sort of how do we quantify it, how that difference might occur by more granular ways of looking within Hispanic Latino individuals. All right, so the work that I'm showing you here is mostly done within the PAGE study. PAGE is the population architecture using genomics and epidemiology. It's a long-running study. I think by now it's about 15 years long going. It was started by NIH in 2008 when they recognized they had a diversity problem, which I think is pretty telling that 15 years ago they recognized they have a diversity problem and we still haven't really done that great by it. And so the people who I'm showing you here is a principal components plot. Each dot is a person and they are colored by how they self-identify. And so on the right-hand side, you have sort of the inferred African ancestry component being pulled out. At the top, East Asian. In the back, sort of this indigenous, the Americas being pulled out. And at the bottom, there is Europe for a variety of reasons in human history. And what you see here is across all of these groups, there's no discrete clustering. It really made no sense when we saw this data and the groups that we were focusing on to possibly stratify our analyses by these labels, because genetically speaking, there's not a lot of discrete categorization you can happen that makes really any meaningful difference. And so that's sort of one of the things that we want to talk about, but I want to focus mainly in this talk about the group that self-identify as the label of Hispanic Latino, which I think I'm a little biased in this fact, but is the most nonsensical label in genetics that could possibly exist. But here we go. So what do we mean when we say Hispanic Latino when thinking about genomic research? So here is the same people from the previous plot. It's rotated a little bit, but these are people who are identified as Hispanic Latino. And what you can see is they have ancestry from everywhere, which is not surprising. There's been many papers, including the people in the room that I saw, who characterize this really, really well. And then also what we see is there is substructure within this group. So if you allow people to identify further, and you'll see the groups in the next slide, is that you do see some substructure, mostly in later principal components, but you see a little bit in the PC1 and PC2 as well, given different demographic histories across the Americas in regards to African, indigenous Americas and European ancestry. And so one of the ways that we conceptualize this for this study and looking to see how ancestry influences genetic risk is to look at admixture proportions. So each line, vertical line, is a person, and they're colored by the proportion of their genome that's inferred to be from these different components. And what you can see on the right-hand side is Hispanic Latino, and there's a large amount of variation within that group. And if you separate it out more granular, people can identify themselves further, you see very big differences on average between these different groups. And I think it's important to note that if I had a single person, I could not tell you from their admixture proportions how they self-identify. But there are differences on average with the groups that are informative to how we model their genetic risk. Okay, so we've built this relationship. We've shown there's a relationship between the sort of group membership and then, group membership and ancestry with the SNP and BMI. So let's see if we can build out this DAG or directed acyclic graph a little bit better. And so what is the relationship between group membership and BMI, the outcome? And so here I'm going to rely on previous studies that have been conducted. We work with collaborators in the Hispanic Community Health Study and Study of Latinos, which is based in the United States. And they have done extensive work looking at the health of Hispanic Latino groups. Here, what I'm showing you is that there's several studies looking at there's differences between groups in BMI, in weight gain trajectory, also in the burden of cardiovascular disease risk factors. And so it's sort of important for us to note that there are differences. This is not really surprising, given that there are different groups with different histories, different environments, but we do have substantial evidence to quantify this as well. Okay, so you have this relationship between group membership and BMI. Now let's see if we can build it up a little bit further. So now I'm interested in seeing, okay, we know that there's differences in performance of a risk score, this relationship between SNPs and BMI. What happens when we compound it in a genome-wide association, or in a polygenic score, which is genome-wide? And what do those dynamics look like within this one group that is often assumed to be homogeneous? And so for this, we applied a polygenic score for BMI to our study participants. And first, I want to show you sort of what the relationship is between the score and these different ancestry components, these admixture proportions. What you can see here on the left-hand side is each dot is a person, and there's a strong positive relationship between ancestry, this AME ancestry, and the score itself. So people who have more of this indigenous ancestry have a higher score on average. And there's sort of two options for why this could be. One is that it's a real relationship. The more ancestry you have, the higher BMI. It's sort of a real thing that we need to account for. Or it's confounding, right? It's just because of different genetic drift of the alleles, and it needs to be controlled for and sort of eliminated from this risk model. And we can actually see that on the right-hand side in the model fit. So this risk score, if you don't adjust at all, has an R-squared of 0.038. It goes up a little bit if you adjust for the top principal components, so I'm sort of throwing everything at it. But what's really interesting is that the model fit actually does the best when you explicitly adjust only for the thing that has the sort of false relationship, which in this case is this indigenous ancestry component. And so it's sort of important for us to note that for this, it's really important to control for the ancestry in a very deliberate way. I think often we see it as a nuisance parameter where you just throw a bunch of principal components in, and sometimes that's not the best for the best model you can get. And so what I want to show you too is what happens if you don't, and sort of the consequences for if you do things improperly. So what I'm showing you here on the x-axis is, again, the proportion of this ancestry. And then on the y-axis, the changing in ranking after adjustment. So if it goes above that dashed line, your rank gets upgraded, so you have more risk after the adjustment. And below the dashed line, you have lower risk after the adjustment. And what you can see is that it pivots, sort of this bow tie effect, around the average proportion of inferred ancestry. So if you're modeling to the mean, but you have a very wide variance in your population, you're doing the most disservice to those on the tails, which you can see here. And so without adjustment, if this score had been returned to individuals without proper adjustment, their risk would have been underestimated if they had low of this ancestry and overestimated for those with the high ancestry. So there are real consequences for improper adjustment for ancestry within a group. And we can look in terms of the deciles. For polygenic scores, we really don't care if you're 50th versus 55th percentile. We just care if you're at the extreme. So very high risk, very low risk. And what you can see here is if we use the top decile at the PRS versus everyone else, and then the lowest decile is low risk. Before and after adjustment, most people stay the same. It's not very surprising, but 5% move up and 5% move down. And you can see in the middle here that those who move up are mostly in the lowest quintiles of ancestry, and those who move down are in the highest quintiles. So this sort of differential bias in who gets overestimation of risk and who has underestimation of risk. That is alleviated if you actually properly adjust for ancestry in a sort of deliberate way. And so if we do this before and after adjustment, you see these increases. Looking at before adjustment in sort of the see-through color and after adjustment in the solid color. But you still have these differences, right? These differences between these group memberships and these large differences in terms of how well this genetic score can capture BMI. And for us, we sort of want to delve in further and say, okay, is this because of some different environmental contexts based on the different groups, the United States? Is this because of differences in genetic ancestry, because there's different proportions of ancestry? How can we sort of quantify this? And so we're going to get more and more of a complete picture here. We just talked about this, and we're going to sort of delve more deep into this dynamic here. And so before, this is sort of what I showed you. This is after adjustment, where you have this different performance of a full model for the risk score that also includes age, sex, and study. And so what you can see here is that there's different levels of performance of this full model between the different groups. But what this sort of is sort of masking, and often what masks this dynamic in risk studies that are published, is that there's a lot of things going into this model here, not just the genetics. So we can actually partition the performance of this model into these two components, right? So one is just the risk score itself, which has already been adjusted for ancestry, so that's sort of been already dealt with, and then the base model itself. And so you see here that there are different proportions. So if we sort of realign things to make it easier to interpret, what you can see here is the proportion of the variance in outcome, so in this case, body mass index, that is explained by the polygenic score. And so you see the highest in the South American group, which I also recognize as an entire continent, but for the sake of numbers was collapsed in these studies that participated in PAGE, and the lowest in the Dominican group. And so you see this very different performance based on this group membership. What I think is also interesting is that you see very different performance of the base model itself, which is just age, sex, and study, where just including age and sex does not explain a lot within the Mexican group here, but it does explain a lot in the Puerto Rican individuals. And so you have this difference in what the model is actually explaining between the different groups and sort of what's going on there. And so we can delve into this a little bit further. When we do modeling in genetic epidemiology, often the default is to just keep on adjusting. So we would see these differential performances, possible different dynamics, and we'd say, okay, maybe it's a confounder. Let's throw it into the model and see what happens. But I think it's important to note that this doesn't necessarily mean a better model all the time, right? Because you don't know if what you're looking at is a real relationship or needs to be adjusted for some sort of confounding. And so what we see here is that if we actually adjust for this group membership further, our model fit goes down, it decreases. And the reason for this is because there's actually a real relationship between these different groups and the risk for being able to predict BMI. And so what you can see here is that, if I'm adjusted versus not in this sort of bar plot in the top left here, the people who have the most difference is the Puerto Rican individuals, right? And you can see this on the bottom here, that their distribution of the risk score itself is shifted. It's shifted over. And it's shifted over partially because of the genetics involved, but also the BMI distribution in that population is also shifted. And so it's a relationship between membership in that group and a higher BMI within this study. And so what you can see here also is that if you go before and after adjustment, and you're looking for this fold increase or this increase in BMI in the top quintile for here, that the benefit for adjustment versus not differs by group. And you see that it sort of gets cut off. But so the answer is not to keep on adjusting. Often in genetics, in these large scale genetic studies, it's approached more as an engineering problem with just putting all these parameters in, throw everything in there. But it's not really the way to do things, especially from an epidemiological standpoint. And so when we also try to differentiate between environment and genetic ancestry, one thing we can do is just look for the proportion of variance explained by both of those constructs. So here what I'm showing you is models. Now here I'm comparing and contrasting models for BMI and then a PRS for height. They're good examples because they show you sort of the differences between a trait with high heritability, so height, versus lower heritability, so BMI. And really what you need to know from these things, I'm going to sort of breeze past them because I'm running out of time, is that when you look at BMI, when you take away this group membership, you lose more of the variation explained than you do if you take away the principal components. So this social membership into a community makes more of a difference when explaining or understanding a person's BMI or the population's distribution of BMI. On the other hand, if you look at height, the genetics, the principal components, explain way more of the variation than actual group membership. And this sort of tracks with the one being way more about the environment and one being way more about the genetics. And we can keep on going and delve this deeper. I'm going to just keep on going because I'm out of time. But the idea, too, is that if we look at what's actually being captured in the score itself, which is based on a bunch of correlations from genome-wide association studies, that also differs between groups and also between environmental variables. And so it matters not only the group membership itself, but also the environmental variables that might be present at different prevalences within that group itself. All right. So here we go, and we have the smoking, so that's part of the model. And now what about more of these structural determinants of health? So these larger scale things. And so social and structural determinants of health, there's these 5 categories from the CDC in the United States. But we're going to talk about mostly social and community context for this. And so there is previous work done within the study I mentioned before that showed that the strongest predictor of moderate and extreme obesity, so BMI being higher, is length of residency in the mainland US. And so in this case, the exposure is basically your exposure to an obesogenic environment, which is the United States, so being the United States. And this was consistent across Hispanic Latino backgrounds. And so we have this relationship between these sort of environmental variables and outcome. And acculturation was not significant. So it doesn't matter if you have higher or lower acculturation, just exposure to the US is a predictor. And so colleagues of mine and I looked at this within the study. And really what you need to know is, again, they recapitulate that this model fit differs by background, and that the actual risk score differs by background as well in terms of its beta. But what I think it's really interesting is if you look at the effect of when an individual immigrated to the United States and how that changes the performance of the risk score, it depends on which group you're coming from. So what you see here is that in those individuals who are South American and they came when they were very, very small children, the risk score does much, much, much worse. See the negative effect size than in those who came as an adult. And then sort of the inverse is true for the Cuban individuals. And this is because of many things, mainly why people came to the United States, when they came to the United States, and where they go when they come to the United States. And so this idea of collapsing everyone together makes very, very little sense. And so, just to say that I know this comes a bit of a downer when I talk to genetics audience, because it gets very complicated and it sort of requires this extensive framework to better understand the health of these groups. And so what does this all mean? So the conclusions for this study is that, I think first of all, that the grouping of individuals from 2 different continents with very diverse histories, cultures, ancestries, environmental contexts makes very little sense when it comes to genetics, and it makes very little sense from an epidemiological standpoint. It's done for convenience, and it's sort of done as the consequences of thinking that stems from hypodescent and eugenics, but it's not really valid in any way. The other thing is that admixed individuals are not just a sum of their parts. This is a common refrain that we have to keep on going through in genetics communities, and that everything is very, very complex. And so we need these sort of integrated models moving forward. And this includes having models not just from the genetic side of things, but really capturing the entire sort of ecological system really for humans. And this includes sort of at the individual level, you go down to the sort of genomic substrate and then up to gene expression and system level approaches. For past that to micro level, meso level, macro level, and global level contexts. And that includes sort of when you look at this racial ethnic context. And so this really makes us question what the default is. We all make assumptions when we model human health, but I think this is very different as to what we accept as the default, which is often what you find in papers in which it's simple language, even that if I say that I work in diverse populations, I think everyone in this room, all right, telling me to be quiet, is that that I'm talking about non-European groups, which I think is a silly thing. And then also just to end here is that when we think about who we do this work for, this is the distribution of the GWAS catalog by population. It is not reflective of our world population. It is not reflective of the US population, but I can tell you what it is reflective of, and that's us, which is very embarrassing. I know I didn't go into this for purely selfish reasons. I'm sure most people didn't either, and so this sort of needs to be helped. And so with that, as we move to this precision health, we need to think about who is it actually precise for. And I have a lot of people to thank for. My lab is called the Rage Lab. And I can take any questions. I think we're out of time, but I'm also happy for emails and messages as well.
Thank you very much, Jen. Super interesting talk, very enlightening. We have time for 2 very quick questions if someone wants to ask something to Jen. Any questions in the audience for Jen? Maria, thank you.
Bueno. Hi, Jen. Thank you.
Hi.
I found it very interesting when you found this pivoting point at the average ancestry of each group. But what's the relationship with the variance in the ancestry? Because these can you can have an average point, but have huge variance. What's the behavior like?
You mean the variance by like Hispanic Latino group specifically, the more granular groupings?
The variance in the proportion of an ancestry, like in this case, Indian or Native American ancestry.
Yeah. So one thing I should note here is that there is it differs by group and it differs by age. I think it's really important to have that sort of footnote within the United States itself, because so here you can see the difference of variation. I mean, one of the examples I think that is really sort of important, and it's not just because I'm married into a Cuban family, but because it's a good example, is that in our study right here, you can see the Cuban individuals have a lot of European ancestry. This is not reflective of genetic trends in Cuba, but rather reflective of who was allowed to leave in larger numbers within this age group to the United States due to different immigration policies, and due to also where the recruitment occurred. And so we see differences between the different groups. I mean, you see the most really within the Mexican American group. But I would just say this sort of disclaimer that it is really a function of sort of who you're sampling and when you're sampling those individuals, at least here in these studies.
Thank you very much, Maria. If there's no other questions in the room, yes, Andres, please.
Hi, Jen. Great talk.
Hi.
Thank you so much for presenting. So yeah, very interesting work where you were considering several variables to model this trait prediction for BMI. And I was wondering if it would be possible to then control for ancestry, but instead of global ancestry or average ancestries per population, the actual local ancestry of those markers considered for the polygenic risk score prediction.
Yeah, I think that's where a lot of methods are going to try to control for this. I think the main challenge we have for creating polygenic scores that are much more focused on local ancestry versus global ancestry is just the lack of source data, the lack of source GWAS, the individual level genetic data to find those sort of local ancestry track effect sizes, because you have much smaller sample sizes on average in these studies. And then if you also split it up by the different, haplotype tracks, then it gets even smaller. So I think with studies like All of Us, which are much bigger here in terms of diverse representation, we can sort of chip away at this. And also with your lovely paper that was published yesterday, also with those sort of data sources, that would also help us move forward in this. But I see that's sort of one of the bigger challenges for that.
Thank you very much.
Thank you very much, Jen. Again, it was a pleasure to have you as the first keynote of our event. And we're looking forward to having you in the future. Thank you again.
Thank you.
Okay.